Action, I think you can start now.
Okay. All right. So today we will do the second class in organogenesis. Based on the feedback that I received from all of you after the last class and some conversations that I had with a few students, I think I spoke with Khushbu and a few others. My sort of impression is that this class should also be directed at a slightly broader overview of organogenesis, um, with a focus on another organ, not the lung. So for reasons that I will go into in the course of this presentation, I decided not to go into the liver, which is something that many of you had requested, simply, I think, largely motivated by the fact that this is an organ that can regenerate and so on. But conceptually, I decided that the liver wouldn't be as interesting to focus on as would be the kidney, simply because of the extent to which you can cross correlate concepts and processes in lung and kidney. So they're very different, and at the same time regulatory principles and so on are similar and different. So I think that sort of suggested to me that we should focus, instead of focus on the kidney rather than focus on the liver or the pancreas. But I'll go into the details of why I made that decision later. So so let's start with a quick recap of what we covered in the last class, defining the sort of domain of organogenesis, and then quickly go over some of the principles and processes that we discussed in the last class in the context of the lung, and then look at the process of kidney development. So so if you look at the development of the mouse, and this is of course a very germ cell-oriented description of the development of the mouse, but I think the stages that it represents are conceptually relevant here. So we start with, of course, fertilized egg, a zygote, which then undergoes multiple rounds of division to yield a morula, which then self-organizes to form a blastocyst, which then implants. And at this point, you've created a distinction between the extraembryonic tissue, the prophoectoderm, and the inner cell mass, which will generate the embryo. This structure undergoes gastrulation, and this process of gastrulation then creates three germ layers: an ectoderm, a mesoderm, and an endoderm. There is a lot of complex tissue reorganization that happens from this simple structure that contains three layers of cells to forming an embryo, which now contains two tubes that run from head to tail, a neural tube and a gut tube. The gut tube, of course, is endodermally derived. The neural tube is ectodermally derived. And I'll have a little bit more to say about this because I think it'll be helpful to you in terms of your future studies on development. And then, of course, the gut tube gets patterned into domains: foregut, midgut, hindgut. And then from each of these domains, there arise organ primordia, primordia for organs like the thyroid, the lung, the liver, the pancreas, and so on and so forth. The remainder of the gut tube, of course, is patterned to form the digestive tract. And the process of organogenesis, as I recognize it, or as many people recognize it, is really marks the transition from a precursor structure, such as the gut tube or the neural tube, or coalesced groups of cells in the mesoderm, and this is something we will again discuss more extensively today, to the formation of the first detectable structures that represent the organ. So organogenesis begins there. In some sense, you could imagine that all of this is organogenesis, but pedagogically, organogenesis is from the time you can first begin to detect a structure in the embryo that is the precursor structure for the adult organ. And most of these processes start post-gastrulation in the mouse, and they start around E8.5 to E9.5 in the mouse embryo. So just a few things. I mean, and I'm only making this comment because I think this sort of broader overview of development will help you in terms of relating to other lectures, both in this class and talks that you may hear outside. I think the one of the places where a lot of students tend to get lost is, as I mentioned, in this complex gymnastics, tissue gymnastics, and reorganization that happens during the early embryo. during early embryogenesis, particularly after gastrulation. You see, the specific gymnastics can vary tremendously across the type of vertebrate you look at, and definitely varies between vertebrates and invertebrates. And this is where a lot of people tend to get lost. So just to keep it simple, if you were to focus on vertebrates and look at them very generally, in going from a structure that has roughly 3 layers of cells, meaning ectoderm, mesoderm, endoderm, to a structure which forms the basis for all organogenesis subsequently, there are 2 things or 3 things that happen, 2 of which are represented here. The third is something that I will discuss in greater detail in the context of the kidney. So the 2 things that are happening is the formation really of 2 tubes that form by the pinching of existing epithelia into closed structures. The first, of course, is shown in yellow here, which is the formation of the gut tube from a field of cells that are of endodermal origin. This gut tube again runs from head to tail. In addition to this, of course, you've got another tube that forms, and this forms from the ectodermal layer. And this forms, again, by a pinching off of an existing epithelial tube to form another epithelial tube, and that is the formation of the neural tube. The other important process, which is important to remember here, is that the gut tube will give rise to all of the visceral organs. The neural tube will give rise to the spinal cord and the brain. During the formation of the neural tube, and Varsha is the local world expert in this matter, there are a group of cells at the cusp of where this sheet that is going to form the neural tube closes, that exit the epithelium, undergo what is called an epithelial to mesenchymal transition. And these cells, of course, are the cells of the neural crest. And these cells, once they leave the ectoderm and the invaginating neural tube, will give rise to all of the peripheral neurons in your body. So if you were to think one step further in terms of organogenesis and how organogenesis initiates along both of these tubes, what you will find, of course, and this is something that we discussed in the context of the lung, that the existing tube will, via interactions with the subepithelial mesenchyme, begin to undergo branching morphogenesis. And in the case of the lung, as we discussed, the mesenchymal cells produce FGF10, which then acts on the epithelial cells to induce directed growth, while simultaneously inducing sonic hedgehog via the ETV4, ETV5 transcription factors, and sonic, then released by the epithelium, represses FGF10. So you have a negative feedback loop which is established, and this negative feedback loop is used iteratively during development to create the entire branch structure. The big picture here, of course, again, goes back to the fact that from an existing tube, you form 2 tubes. An existing epithelial tube gives rise to 2 epithelial tubes. And of course, in the context of the central nervous system, all of the brain and the spinal cord is derived from a tube or consisting of a single layer of cells. And cells within this tube can either divide symmetrically or asymmetrically, and then delaminate from this tube via, again, a process of epithelial mesenchymal transitions. And these cells then exit from this tube where the apical margin or the apical boundary is shown here, migrate into the tissue itself, and then give rise to an entire population of neurons. So your entire brain forms basically by this manner. Derived from a single tube, cells divide symmetrically or asymmetrically to create neurons, glia, progenitor cells versus more descendant cells. These cells then exit, migrate into the lamina, and differentiate. The reason I took all of this time to describe this to you is because the kind of tube morphogenesis that we will describe today is very different from either of these 2 processes or what happens subsequently, either in the context of the lung or in the context of the brain. The kind of tube morphogenesis we will discuss today in the context of the development of the kidney starts with a group of mesenchymal cells, cells that are loosely packed without any obvious cell-to-cell junctions, and then undergo a process called a mesenchymal to epithelial transition to create an epithelial sheet that then undergoes multiple rounds of branching and so on. So just a quick recap of the lung, and I'm going over this simply because it's also going to be relevant when we talk about the kidney, and it sort of helps you to make connections about processes and concepts. The word lung is synonymous with the respiratory tract, and it's composed of airways and alveoli. But in fact, the lung is an organ consisting of many different tissues. For example, the epithelial cells of the airways and alveoli are endodermally derived. The airway smooth muscle which surrounds the airways is mesodermally derived. The blood vessels and the capillaries, the smooth muscle that surrounds them, are mesodermally derived. And of course, the The lung contains neurons. Again, these are neural crest derived, or they contain axons that innervate them from the central nervous system. And all of these neurons are, of course, ectodermally derived. So the lung is an organ consisting of many different tissues. Each of these tissues has a different developmental origin, and the development and repair of these tissues is dependent on different populations of stem cells. Quickly to recap, the process of branching morphogenesis in the lung is highly stereotyped. This is going to be very important because in the kidney, as you will see, it is not highly stereotyped. So what that means in terms of the branching pattern being stereotyped is that in 3 dimensions, both in space and time, branches have the process of branching follows exactly the same pattern. Meaning you can take 2 different lungs from 2 different animals at the same stage, and you will find that they have virtually overlapping patterns of branching. And of course, this entire branch network is generated by 3 different branching subroutines: domain branching, planar bifurcation, and orthogonal bifurcation. And the order in which these branching subroutines are used to create a branch structure like the lung, sort of visually shown here. The initial branches are all via domain branching. The later branches are essentially a function of either planar bifurcation or orthogonal bifurcation. We will come back to this pattern when we discuss the mechanisms and the modes of branching morphogenesis in the kidney. Of course, this branched tree creates a structure of, this branch tree essentially creates the basis for the airways. As you know, the lung contains 2 components: the airways, where the gas, which is for the exchange of gases, and the alveoli, which is for the passage of gases, and the alveoli, which is required for the exchange of gases. The development of the alveoli occurs after the establishment of the branched airway tree, and the way in which the growing structure switches from making airways to making alveoli involves a systemic pulse of a glucocorticoid. Right? Glucocorticoid signaling is activated, and this leads to the cessation of what is an airway genesis program in a group of cells that are at the distal tips of this airway tree, which express SOX9. And as a function of glucocorticoid signaling, these SOX9 positive cells stop activating SOX2, which is the genesis of an airway program, and allow the cells then subsequently to undergo morphogenesis to create the alveolar network. So in other words, you have a singular branch tree with a group of cells at the tip that in time switch from making airways to making alveoli. Again, this pattern is going to be extremely interesting and to compare with in the context of the kidney. Finally, we come to the process of cell plate specification. Cell plate specification in the lung always happens, follows the process of branching morphogenesis. So once the distal tip has grown, the cells that lag behind gradually activate signaling pathways like Notch, which then leads to the lateral segregation of cell fates to making the cells that comprise the airways. And the same process is used again in the context of the alveolar epithelium to subdivide the alveolar epithelium into alveolar type 1 cells and alveolar type 2 cells and so on. So if I was to condense all of what we discussed in the last class into two different bins, or put all of it into 2 different bins, I think this is what we would find. I think if you think of organogenesis in the most general terms, you have core processes and you have core concepts. If you look at the core processes, there are 2, and these are sort of temporally staggered, of course. Morphogenesis precedes cell plate specification. And in the process of morphogenesis, you have tube morphogenesis. In the context of the lung, this involves the genesis of the gut tube and its subsequent refinement to the foregut, midgut, and hindgut. Following tube morphogenesis, you have branching morphogenesis, which is where an existing tube then undergoes an iterative process of branching to generate a branch network. There are changes in tissue structure that occur during branching morphogenesis. This is not very important in the context of the lung, specifically in the context of the respiratory tract, but it is relevant to the lung. For example, the 2 types of changes in tissue structure that do happen are mesenchymal to epithelial transitions and epithelial to mesenchymal transitions. As I mentioned, these 2 are not very relevant in the context of the respiratory tract, but they do play a role in the genesis of other aspects of the lung. For example, the EMT is very critical to the process by which neurons enter the lung. The mesenchymal to epithelial transition is particularly important in the context of the pleura, but we will discuss that later, or if at all. And finally, cell migration. There are 2 types of cell migration that are important to organogenesis. One is epithelial migration, and this is something that we didn't talk about, but has been beautifully described in the lung. Well, even cells that are within the epithelial layer can migrate within the epithelial layer to form clusters of specialized cells within the epithelium. This is responsible for the genesis of a structure called a neuroepithelial body in the developing lung. And of course, there is the migration of mesenchymal cells, where cells which are at the distal tips, for example, will migrate subsequently along the sides of the tubes and give rise to the smooth muscle that surrounds the airways. And then following morphogenesis, you have cell fate specification. The core concepts, I think, which are relevant to these processes that occur during organogenesis in the lung is that morphogenesis is stereotype, meaning, again, in 3 dimensions, the position, the pattern of branching of the airway tree is completely stereotyped from animal to animal. Yes, there are some differences across strains. Yes, there are some errors during the process of branching, but overall the process is stereotyped. Second, epithelial and mesenchymal signaling and crosstalk is very important. As I discussed in the cartoon previously, FGF10 signaling from the mesenchyme to the epithelium, sonic signaling from the epithelium to the mesenchyme, and this negative feedback regulation is very, very important in the context of driving branching morphogenesis. What do these signals do? These signals basically establish gene regulatory networks, which establish stable patterns of gene expression and allow cells to either maintain the fate that they're in or allow them to transition from the fate they're in to becoming more specialized. Stem cells are involved. This is an extremely important concept. It implies, of course, that the entire branched network that forms and all the cells that comprise this network are derived from a population of cells that sit at the distal tips, which can both make themselves and generate cells that then differentiate to form the different cells of the organ. And last, I've circled this because this is a part, I think, or an aspect of vertebrate organogenesis, which is very poorly understood. It speaks also in part to the question that Utkarsh asked in the last class. And that is that the dynamic modulation of cell-cell and cell matrix interactions is going to be extremely important in driving this entire process. But our understanding of how this occurs in the context of vertebrate organogenesis is rather poor, arguably best understood in the context of the limb bud, and that is something that I think we will hear about subsequently, but not very well studied in the context of most of these organs that we frequently describe, like the lung, the liver, the kidney, and so on and so forth. All right, so this brings me to the focus of today's class, which is the kidneys and the urogenital system. So let's start with what we used to study in our class 10 textbooks, just understand what the kidney is, what it is made up of, how it works briefly, and then go into understanding how it develops. So the kidney basically consists of two compartments or lobes, if you will, the left kidney and the right kidney. Its principal function is to filter blood. In other words, basically remove impurities from blood, maintain water homeostasis, and basically of total volume of several liters of blood that flows through our body, we are producing a small fraction of that in the form of urine every day via the process of filtration that occurs in this particular organ. So what this organ contains and must contain at some level, as you might imagine, is a very rich supply of blood vessels, the renal arteries and the veins, which bring the blood to this organ and transport the blood from this organ. In addition to that, what this organ must contain is a filtration unit, and that filtration unit is basically the nephron, which then allows for the selective movement of materials between the blood and the collecting system, which is the third component of this organ, which then contains all of the solutes and the water from the blood, and then transports this content, which is essentially the urine, from the kidney via the ureter to the bladder, and from the bladder via the urethra, disperses the urine. So if you think about how this entire process works, well, it ultimately comes down to or converges on what is the glomerulus, which is the specialized structure that you see here in the nephron. Basically, there is a basement membrane, which is a layer of extracellular matrix, which serves as a selectivity filter for the content of the blood, so that only select components can move in and out from between the inside of the blood vessel and the collecting ducts, which comprise the glomerulus. So to put it briefly, the kidney contains two aspects: a cortex, which contains all of the filtration units, the nephrons; the medulla, which basically contains the collecting ducts, and a rich supply of blood vessels, which is basically the renal artery that undergoes a series of branching steps to make a very fine mesh of capillaries, which then sits inside the glomerulus. So all of the blood coming in first passes through the glomerulus, which sits in the cortex of the kidney. And this filtration then leads to the collection of urine, which by a series of tubes converges into larger and larger and larger collecting ducts, which then transmits the urine or transports the urine into the bladder, which lies somewhere here. So this basically is, in a nutshell, the structure of the kidney. And now what we're going to discuss is how this network is formed. So again, just some nomenclature, which I think is important. Generally, the word kidney is synonymous with the urinary tract that is composed of nephrons and collecting ducts. But in fact, the kidney, again, is an organ consisting of many different tissues. So the epithelial cells of the nephrons and the ducts are mesodermal in origin, unlike in the lung, where they are endodermal in origin. This is a cut and paste problem. There are blood vessels and capillaries, which are mesodermal in origin. There are nerves that are ectodermal in origin. And of course, the ureter that comes out of the kidney, which is basically the large collecting duct, which collects all of the urine that is accumulated in the kidney, feeds into the bladder, and the bladder then connects to the urethra. And so the bladder and the urethra in the context of the urogenital system is endodermally derived, not mesodermally derived. So in toto, the urogenital system is both mesodermally derived in the kidney and endodermally derived. So let's now get into the process by which you make the kidney. So as I mentioned, when we talk about organogenesis in the kidney, we typically focus again on the urinary tract, which contains two components. It's basically a single epithelial sheath, which is highly branched, that contains two components. The first is the component that sits in the periphery, which is the nephron. And the second is a component which is connected to the nephron, which is a series of collecting ducts. And these collecting ducts basically tend to be concentrated in the medullary region. The nephron actually extends from the cortex into the medulla, but that's just a minor detail. What is important here is to sort of compare the branch structure of the kidney to the branch structure of the lung. So the branch structure of the kidney, again, now I'm going to reverse the order, consists of a series of collecting ducts, which meet up with a series of nephrons that are present in the periphery. The branch structure in the lung consists of a series of airways, which culminate in a series of sacs in the periphery, which are called alveoli. The principal difference, arguably the most important difference in the nature of the processes that build the urinary tract and build the respiratory tract in the lung, is the fact that the branch structure in the kidney actually is derived from two completely different populations of cells. In other words, the collecting ducts and the branch structure of the collecting ducts originates from one population of cells. The nephrons originate from a completely different population of cells, and we will see what those populations are and so on and so forth. And then finally, these two are stitched together to make the entire urinary tract as relevant to the kidney. The respiratory tract, of course, as we've already seen, contains an epithelium which is derived from a singular population of cells that gives rise to both the airways and the alveoli. So let's start looking at how you would make this branched structure. And then finally, after we finish this description, we'll go back and ask the bigger questions about how stereotyped it is, and so on and so forth. So the origin of the kidney is happens around the same time as the lung. It starts around E9.5. But contrary to how the lung and the brain develop, the kidney originates from mesoderm. In other words, it originates from a group of cells that have no obvious cell-to-cell connections or no defined cell-cell junctions. So what happens at this stage, and I'm going to simplify this tremendously because actually the process is way more complicated than this, is that a group of mesodermal cells tend to sort of aggregate together to form what is called a core. The nature of the cell-cell junctions in this sort of aggregate of mesodermal cells are not very well understood. In some cases, the N-cadherin, for example, has been implicated in the formation of these clusters. But really, the forces and the molecules that hold these cells together to form cords or cores, if you will, not very clearly understood. Nevertheless, there is a group of mesodermal cells that cluster together to form what is called an intermediate mesoderm. And the location of the intermediate mesoderm is sort of shown in this figure. This is the neural tube. The gut is somewhere, the gut tube is somewhere here. This is the notochord. And what you're seeing here is the intermediate mesoderm. So this intermediate mesoderm, which basically again consists of a cord of mesodermal cells that are sort of loosely packed together in some manner, then bifurcates into two components. It bifurcates into a structure which is more relevant here called a nephric duct. This nephric duct undergoes what is called a mesenchymal to epithelial transition. In other words, these are a group of mesenchymal cells which self-organize or reorganize to form an epithelial sheath. And this epithelial sheath also develops a lumen. So what is happening in the context of this developing embryo is that as the embryo is extending, meaning there is extension of the embryo from the cranial to the caudal axis, the intermediate mesoderm is also extending and growing in size, and the cells of the nephric duct in particular are undergoing a reorganization to form an epithelial tube. This process of reorganization, again, involves epithelial to mesenchymal interactions in the sense that these cells are actively interacting with the cells that are neighboring them. And it involves the establishment of a gene regulatory network, which then leads to a stabilization of expression in the nephric duct. So there's the complexity of kidney development, and this is the reason why most people who look at or try to understand kidney development by reading textbooks or by watching videos get completely confused, is that the process of kidney development in a vertebrate is actually a three-part process. See, the vertebrate, like a mouse, tries to make the kidney 3 times, and 2 of those 3 occasions are largely failed attempts. The reason why the mouse has this very unusual program of kidney development is probably of evolutionary significance. And we can discuss this later. But the point is that there are 3 different phases of kidney development. There's a phase which we call the pronephros formation, phase which is called the mesonephros formation, and finally a phase called the metanephros formation, which is really the time at which the residual intermediate mesoderm and nephric duct begins to form the adult kidney. So once this once this nephric duct is formed, the distal tip of the intermediate mesoderm, which is the part of the intermediate mesoderm which is really closest to the tail, then undergoes this process of reorganization, leading to the establishment of what is called a metanephric mesenchyme. This is again, remember, all of these are of mesodermal origin. These cells are mesodermal cells that have undergone a mesenchyme to epithelial transition to form an epithelial tube. These are mesenchymal cells that begin to aggregate in some manner. They are not anatomically distinguishable, but they're distinguishable in terms of patterns of gene expression. And what this then leads to is the thickening of this epithelium, or the pseudostratification of this epithelium, meaning there are more cells that begin to pack per unit area. And this region then begins to bud out to form what is called a ureteric bud. This ureteric bud then intimately interacts with this metanephric mesenchyme that forms a cap around it. And there is a very intimate interaction between these 2 structures that then leads to both the branching of the ureteric bud and to the proliferation of cells that constitute the cap mesenchyme. And this process then repeats itself over several generations, over a period of 5 days, to give rise to this highly branched structure of collecting ducts that form. So what you can see here, for example, is a cast of how the process of ureteric bud formation and collecting duct formation happens in a developing kidney. So there is an initial process of branching, and then this branch structure undergoes a repeated program of branching morphogenesis to yield this entire structure. So remember, what we're doing here is starting from a cord of cells that has undergone an mesenchyme to epithelial transition. The distalmost part of this cord, which is called the Wolffian duct or the nephric duct, then interacts with a group of mesenchymal cells that are right next to it. And then signals are exchanged, and we'll get into what those signals are. And these signals lead to the branching morphogenesis of the nephric duct to form what is called the ureteric bud. And these ureteric buds then undergo a repeated series of branching via reciprocal interactions with the cap mesenchyme that surrounds it. But all of this is making the collecting duct system of the kidney. We haven't come to the nephrons yet. And if you will remember, I've already told you that in the context of the kidney, the collecting ducts and the nephrons, while they are part of the same branch structure, have different developmental origins. So let's speak to how the process of ureteric bud formation happens during kidney development. And what you can see here is an outline of the epithelial to mesenchymal interactions that happen during this process. The key driving signal is GDNF, which is glial-derived neurotrophic factor, that acts on its receptors, RET and GFR alpha, to lead to a series of changes in gene expression, including the expression of these transcription factors ETV4 and ETV5. And you will recognize these earlier from our studies on the lung. And what happens is a function of the activation of GDNF signaling in the ureteric bud is the production of Wnt11 in the ureteric bud. And this Wnt11 then is secreted by the ureteric bud, acts on the mesenchyme to promote the expression of GDNF. So the point I want to make here is that much like we saw in the context of the lung, where there are reciprocal interactions between the epithelium and the mesenchyme that surrounds it, there is a similar crosstalk that is driving ureteric bud formation. That crosstalk involves GDNF primarily, which is secreted by the mesenchyme. It involves activation of its receptors RET and GFR1, the activation of RTK signaling. And in this case, RTK signaling leads to the transcriptional activation and then secretion of Wnt11, which acts not in a negative feedback loop like we studied in the lung, but in a positive feedback loop to stimulate GDNF expression. What you will also realize from this figure is that in addition to GDNF, which is the primary signal that is produced by the metanephric mesenchyme, leading to the budding and the branching of the ureteric bud, there are other signals, and one of these is should be extremely familiar to you by now, and that is FGF10, which acts in some minor capacity to activate its receptor FGFR2, and then again converging on these transcription factors ETV4, ETV5. So the reason why people discovered that there are multiple signals that are converging on the ureteric bud is because what people found is that if you mutate GDNF and RET or GFR alpha, then you basically see kidney agenesis, meaning the ureteric bud ceases to bud and grow forward. But if in this mutant background, you choose to remove a negative regulator of RTK signaling, which is protein, then you can partially restore branching. And this is what led people to discover that in addition to signals like GDNF and RET GFR alpha, there must be other signals, arguably, which are acting via a receptor tyrosine kinase-based signaling pathway to regulate branching morphogenesis. And so what they then noticed is that if you made a quadruple mutant mouse, where you removed glial-derived neurotrophic factor, you removed RET or GFR1, you removed sprouting, and you removed FGF10, then again you would see no branching at all, and you would see complete kidney agenesis. So unlike in the context of the lung, where there seems to be a very simple hierarchy that is driving the entire process of branching morphogenesis, and that involves FGF10 and FGFR2, in the case of the kidney, there is a primary signaling pathway, which is GDNF and RET. But in addition to that, there is a secondary signaling pathway that can compensate, and that involves, again, FGF10 and FGFR2. Finally, downstream to these signaling pathways, in the kidney, there is Wnt11, which acts in a positive feedback loop to maintain GDNF expression. So this sort of speaks to this concept of epithelial mesenchymal crosstalk being an important driver of kidney morphogenesis. So I'd like to go a little bit deeper into receptor tyrosine kinase signaling and its modulation, because I think when it comes to thinking about how these branch structures are forming, it's important to get into some measure of detail in terms of the regulation of RTK signaling. So as you know, RTK signaling can happen by various, RTK signaling occurs by many different arms. One of them, of course, and the most celebrated arm is the RAS axis. The other, of course, is the PI3 kinase axis. The reason I want to go into this particular signaling pathway, and more importantly, all of the regulatory elements of this pathway. For example, downstream to the activation of the receptor tyrosine kinase pathway, either through the PI3 kinase or through RAS-ERK signaling, is the induction of expression of many negative regulators of the pathway, Sprouty being one of them. And this process of autoregulation is extremely important in the context of how branching morphogenesis occurs. So for example, if you were to think about the bud, you would realize that this epithelial sheath, which is undergoing budding, where there is active proliferation occurring, has to do two different things. It has to extend in one direction, and it simultaneously has to increase circumferentially to increase the size of the tube as the structure is growing. So initially, the buds are very small, but then as they undergo repeated rounds of branching, the buds that are left behind increase in size to account for the overall growth of the organ. Now, how this seems to be regulated in the context of branched organs involves the negative regulation of ERK signaling. So what seems to be happening both in the case of the lung and in the case of the kidney is that you're altering the axis of growth of the primary bud by altering the orientation of the mitotic spindles of the dividing cells in the bud. In other words, when you're looking at bud extension, you're seeing the alignment of mitotic spindles parallel to the direction of growth. When you're seeing bud expansion, meaning circumferential growth of the bud, you seem to be finding that the mitotic spindle of these cells is now oriented not parallel to the direction in which the tube grows, but perpendicular to the direction in which the tube grows. And this difference in the orientation of the mitotic spindle, meaning why is it that at the distal tips of these tubes where the structure is growing in length along the axis of growth, do you see mitotic spindles aligned with that axis, whereas further away from the distal tip of the tube where the structure is growing circumferentially, you begin to see mitotic spindles now aligned perpendicular. The reason for this difference, it turns out, converges on the relative activation of ERK signaling. And so how, for example, the levels of ERK signaling or the spatial restriction of ERK signaling, meaning tip versus the trunk of the growing tube, converges on the orientation of the mitotic spindles in these two domains is an open question. This is something that people don't really understand yet. But what most people do accept is that it's most likely not going to involve direct interaction between the ligand and its receptor, but is going to involve more complex regulation within cells in terms of feedback regulation. For example, the levels of Sprouty, the levels of other self-inhibitory elements of the receptor tyrosine kinase signaling pathway that are induced in these different domains of the growing tube. So I just wanted to go into that level of detail because I think it's sort of an important concept to understand in terms of how branching morphogenesis occurs and how a tube decides to grow in length in one direction as opposed to grow circumferentially and broaden, increase the size of its lumen. Right. So what we've covered so far is a process by which epithelial tube of mesodermal origin interacts very intimately with a mesenchyme that's surrounding it to undergo a series of branches to form a network of collecting ducts. This is only one half of the kidney, the urinary tract in the kidney. Other half of the urinary tract in the kidney is, of course, the nephron. So now what we're going to just sort of briefly go over is how is it that the nephron is formed during this process of branching morphogenesis. So it turns out that if you look at the ureteric bud, which is shown here in blue, this of course is the nephric duct or the Wolffian duct, and this is the metanephric, shown in red is the metanephric mesenchyme. What we've discussed so far is this cap of cells that basically constitutes the metanephric mesenchyme, and these are the cells that are going to produce GDNF. There's a very intimate interaction between these two cells, so that both the cells here are proliferating. The ones at the distal tip, as I mentioned, are proliferating in this axis. The ones that are further behind are proliferating in this axis to lead to the increase in circumference. So as this process splits, as this sort of bud splits into two, again, each of those buds is going to be associated with its own cap mesenchyme and so on and so forth. So what is happening? So this process starts at about E9.5, E10.5 in the mouse, right? Now, as this process is happening, there is a process of refinement or specialization that begins to happen in the cap mesenchyme, such that by about E11.5, E12.5, a group of cells in the cap mesenchyme now begin to coalesce together. Again, mesenchyme to epithelial transition is what is occurring, where a group of cells begin to coalesce together to form what is called a pretubular aggregate. This structure then undergoes a complete epithelial transition to form what is called a renal vesicle. This renal vesicle then undergoes a series of structural transitions called a comma-shaped body or an S-shaped body to give rise to a ductal structure, which then refines to forming the glomerulus. So at the time when this structure is formed, you begin to see during development angioblasts, which are, for example, the progenitors of the vascular network, come in, begin to integrate into the structure and give rise to the capillary network that then surrounds or constitutes a part of the glomerulus. So you will remember from the nephron that the nephron basically contains a series of capillaries that are surrounded by the glomerulus, constituting what is called the Bowman capsule. And so the blood flows through the capillaries, exchanges materials, and then these materials are transported through a series of tubes into the collecting ducts, which then form the urine and exit the kidney. So again, this process by which these cells begin to proliferate and cluster together to form an epithelial sheet is induced by Wnt9b, and this Wnt9b is produced by the cells of the ureteric bud. So very intimate interaction between the ureteric bud and the metanephric mesenchyme. The ureteric bud is producing Wnt9b, which is driving the proliferation via local regulation of, again, Wnt4 and FGF8, the group of cells that will constitute the precursors of the nephron. And simultaneously, the cap cells in the metanephric mesenchyme are producing GDNF, which are driving the proliferation and the growth of the ureteric bud. So this is sort of the reciprocal interaction that happens. And I've sort of mentioned that here or shown that here in the context of this particular schematic. Again, Wnt9b is produced by the cells of the ureteric bud. And this speaks to the cells of the mesenchyme to lead to the growth of cells that express SIX2, and we will get to what SIX2 is and what its relevance is. And this is what drives the process of renal vesicle formation. So let's just take a step back and try to visualize what's happening here so that we're completely clear. So you've got this tube which is undergoing a series of branches as a function of an intimate interaction with a group of mesenchymal cells that associate with the cap. And while this process of branching is ongoing, some of the cells that are a part of this cap are segregating from the rest to form a pretubular aggregate. This pretubular aggregate is then undergoing a further reorganization to form an epithelial structure, which is called a renal vesicle. Remember, the branching process or the process of collecting duct branching is still ongoing while this process of renal vesicle formation is happening. So in some sense, the process of collecting duct branching morphogenesis and renal vesicle formation are sort of occurring simultaneously, if you will. Again, very different from the lung, where airway happens first and alveoli, the formation of the alveoli happens next. And then this renal vesicle, which is epithelial in nature, undergoes a series of shape changes, comma-shaped body, S-shaped body, and so on and so forth, to create an epithelial tube, which will then subdivide into many parts to form the different components of the nephron, namely the glomerulus, the ascending tubule, the descending tubule, and how this, and the connecting duct to the collecting ducts of the kidney. Okay, so one of the most important processes that we have encountered in the context of the organogenesis of the kidney, which we didn't really encounter in the context of the lung, is this process of mesenchyme to an epithelial transition or an epithelial to a mesenchymal transition. Most of you will have heard of these processes in the context of cancers and the propagation of cancer cells. But what you perhaps would not have appreciated is how fundamental they are to driving development. So for example, if we take a step back and go right to the beginning, the process by which, for example, the epiblast is formed from the cells of the inner cell mass involves a mesenchyme to epithelial transition. A group of cells that are sort of loosely packed then come together to form a sheet of cells. the mechanism by which, for example, the cells of the mesoderm undergo a structural change leading to the epithelialization of the nephric duct or the Wolffian duct, the structure from which the kidney is formed, involves a mesenchyme to epithelial transition. Epithelial to mesenchymal transitions, for example, best studied in the context of the neural crest, where, for example, a group of cells uh, at the point at which the neural tube closes undergo an EMT and then migrate to different parts of the body to create peripheral neurons. So it turns out that the mechanism by which EMTs occur are relatively better understood than the mechanisms by which METs occur. This is actually, I mean, people have identified signaling pathways that are important for the specialization to occur. People have identified transcription factors that are relevant to MET occurring in the context of a specific tissue. But nobody really understands at any deeper conceptual level what are the cell biological changes that drive these transitions. In the context of the EMT, for example, we know that there are many signals like TGF-beta, Wnt, Hedgehog, Notch, BMP, RTK. There are transcription factors. Snails are very important. Snail1, 2, ZEB1, 2, and of course Twist, which are critical to the process by which an epithelial to mesenchymal transition occurs. But there is no similar conserved collection of either signals or transcription factors that have been identified till date that drive the mesenchymal to epithelial transition, which is sort of integral to kidney development, both in the context of the collecting duct formation and subsequently in the context of the renal vesicle. So the next thing that must happen in the context of any branched organ, if you go back to the lung, you remember that we first talked about the genesis of the tree, the genesis of the alveoli, and then the gradual specification of cells that occurs in the context of the airway tree and in the context of the alveoli. Similarly, the process of sulfate specification in the collecting ducts and the nephrons occur much after the morphogenesis has occurred. The collecting ducts, for example, contain two types of cells. They contain principal cells and intercalating cells. The intercalating cells come in two different flavors, alpha and beta. The process by which the cells in the collecting duct become specified to one of these fates, much like in the lung, at least in the airways of the lung and the alveoli of the lung, involves Notch signaling, which involves localized signaling between epithelial cells, leading to the assignment of either one fate or the other. In the context of the developing lung, Notch signaling is required to specify secretory cells and balance the proportion of ciliated and secretory cells. Similarly, to balance the proportion of alveolar type 1 and type 2 cells. And in the context of the kidney, Notch signaling is required to balance the proportion of principal cells, which are the predominant population, and intercalating cells. So once this branch structure is formed, there are local cell-cell interactions, which involve signals like Notch and Hippo as well, the Hippo pathway as well, but Notch principally, which then regulate the balance of cell fates in the context of this, of the epithelial lining of the branched network. So now we come back to the big questions. And the big question in the context of the kidney, with respect to the lung, is the architecture of the urinary tract stereotyped? So the word stereotyped here requires some definition. So what Ross Metzger showed many years ago when he studied the branching morphogenesis of the lung is that if you were to look at the three-dimensional position of every branch that is forming in a developing mouse lung in time, that you would find that its spatial location in one lung is identical to its spatial location in another lung. So in other words, branches are forming in the same place at the same time, leading to a structure which is virtually identical in one animal and another. Of course, Ross did point out that there is some give, meaning if you look very carefully at the timing of the branching process, what you will see is that there may be in some animals a slight delay in the formation of some branches versus others, but they catch up ultimately. You may find that in some branches there are errors, meaning stochastic losses. So instead of going from one branching subroutine to another, it completely skips the intervening subroutine and goes directly to the final subroutine. So these things do happen. And of course, he also pointed out that across strains, there can be differences in the extent of branching along specific branches. But overall, the pattern is stereotyped. Now, people in the last couple of years, and this work started in about 2013, culminated in a series of studies between 2015 and 2017, have shown that the kidney does not obey these rules. What that means basically is that if you looked at the formation of the collecting ducts in the kidney, you would find that the position of the collecting duct or its branching at any given point in time is not the same in one kidney versus another. And so what this means is that the branching morphogenesis in the kidney, unlike in the lung, is not strictly stereotypic. But what people have done subsequently is to look at higher order aspects of the branch network. And what they find is that even though the branching in the kidney is not strictly stereotypic, there are some features that suggest that in fact the pattern of branching in one kidney and another is very reproducible. What does that mean? So what you can see here, for example, is a color coding of the branch network in a single lobe of the kidney. What people have shown is that the branching within the single lobe of the kidney can be divided into 8 clades, and each of these clades is sort of shown in color. And that while the precise 3-dimensional location of each of the branches within a clade is not the same across kidneys, the extent of branching and the pattern of branching is the same. So this is a slightly more sophisticated idea in terms of stereotypy in branch networks. So what people are saying is happening in the context of the kidney is that in each clade, the branches that form in terms of the number of generations of branching that occurs and the relative balance of these branches, meaning some branches will go on to making many branches while other branches will only make some. This overall, the extent of branching, meaning number of times branching occurs, and the overall balance of branches within each clade is actually very similar, even though the precise 3-dimensional position and the timing of these branching events may not be the same across kidneys. And so what emerges from this analysis is that the architecture of the collecting ducts within the kidney is not stereotyped in 3 dimensions in space and time, but there are aspects to the branching which are highly stereotyped across kidneys. So the other major concept that one has to address when one thinks about branch network like the kidneys. This, by the way, this type of 3-dimensional organization has not yet been taken to the nephron. So in other words, we don't really understand whether the pattern of nephron generation with respect to each of these clades also obeys the same rules. So that is, I think, studies that are ongoing. Right. So moving on, in terms of the concepts that are driving branch networks, one of the key concepts that is driving branch networks, of course, the genesis of branch networks is stem cells and their maintenance. And so one question that you could ask in the context of either the collecting ducts or the nephrons is, are these epithelial branch networks derived from singular population of stem cells? And the indication is that they are. In other words, the cells of the collecting duct, which are derived, from the ureteric bud, seem to be derived from a population of SOX8, SOX9 positive cells that sit at the tips of these ureteric buds. Similarly, the cells that constitute the renal vesicle, which will make the nephron, seem to be derived from a group of 6-2 positive cells that sit in the cap mesenchyme that surrounds the ureteric bud. So in other words, all of the cells that comprise the collecting ducts and all of the nephrons in the kidney seem to be derived from 2 distinct populations of stem cells, one of which is SOX8, SOX9 positive in the ureteric bud and 6-2 positive in the cap mesenchyme. So with that, I'll sort of come back to the place where we started and try to set in context what we have studied in the kidney and how that is different from what we've studied in the lung. So let's go over the core processes. The core processes are morphogenesis and cell fate specification, I think the processes of sulfate specification in both the lung and the kidney are going to be very similar in that they're going to largely involve local interactions, cell contact-based interactions, signals like notch, which then leads to the specification of cells, both in the collecting ducts and in the nephron. I think the major point of divergence between the lung and the kidney lies in the mechanism of morphogenesis. So in the case of the lung, we have an epithelial tube, which by a series of branching events generates a network or a network of epithelial tubes. There is very little change in tissue structure that happens during this process of branching morphogenesis. In the kidney, on the contrary, you have, in some sense, an epithelial tube which is giving rise to a network of epithelial tubes, in that the ureteric bud is giving rise, which is epithelial in character, is giving rise to a network of ureteric buds. But this epithelial tube, which comprises the nephric duct from which the ureteric bud forms, is in fact derived from a group of mesenchymal cells which undergo a mesenchymal epithelial transition to generate an epithelial sheath. More importantly, the entire network of nephrons that are formed are not formed from an epithelial sheath, but formed de novo from a group of mesenchymal cells, again, which sit in the cap mesenchyme, undergo an MET to give rise to that epithelial structure. Cell migration, there is cell migration which occurs in both organs, both in the context of epithelium and mesenchyme, but they are not relevant to the process by which the epithelial lining of either the lung or the epithelial lining of the urinary tract are formed. So I don't think this is going to be an extremely important process. I think there is going to be an important component of mesenchymal migration in the context of the kidney when one thinks about how the cap mesenchyme redistributes itself every time a ureteric bud forms, but these processes are not very well understood. Core concept. In the lung, we studied that morphogenesis is stereotyped. This core concept breaks down completely in the context of the kidney. Yes, aspects of the morphogenesis are stereotyped, meaning within each branching clade in the lobe of a kidney, the pattern of branching and the extent of branching is going to be stereotyped. But the precise location of branches in 3 dimensions in space and time, leading to an identical structure, this aspect of stereotypy is definitely not stereotyped. Epithelial mesenchymal signaling and crosstalk is important. I think this is extremely important in the context of both the lung and the kidney. In the lung, we've studied that it's an FGF10 signal from the mesenchyme that drives branching, involves the production of sonic hedgehog, which in a negative feedback loop suppresses FGF10 expression, and that this negative feedback loop is integral to branching morphogenesis. What we've seen in the context of the ureteric bud is again, an evidence for epithelial mesenchymal crosstalk, where you've got GDNF, which is produced by the metanephric mesenchyme, which activates RTK signaling, leading to the production of Wnt11, which in a positive feedback loop then regulates GDNF expression. In the context of nephrons, again, we've seen that there is inductive signaling between the epithelium of the ureteric bud and the cap mesenchyme, and that involves a signal Wnt9. Wnt9 production from the epithelium is required for the maintenance of the progenitors that will create the renal vesicle and for their subsequent proliferation. So these signals then lead to the establishment of gene regulatory networks in the context of each of these tissues, which is maintaining stable patterns of gene expression during organogenesis. This is integral. This is true for both the lung and the kidney. Stem cells, yes, we studied in the context of the lung that at the distal tips of the growing tree, there are ID2 positive cells, and these ID2 positive cells will sequentially give rise to all of the cells that comprise the airways, and then to the cells that comprise the alveoli. In the context of the kidneys, what we studied is that there are 2 clearly different populations of stem cells that will give rise to the collecting ducts and the nephrons. In the context of the collecting ducts, there are SOX8, SOX9 positive cells. In the context of the nephrons, there is a population of 6-2 positive cells. Finally, dynamic modulation of cell-cell and cell matrix interactions. This must happen in the context of both the lung and the kidney, but an elucidation of exactly what these interactions are is something that that will still require more experimental innovation and so on and so forth. So with that, I'll stop. And I just want to go back to one other comment that was made in the last class, which I think is an extremely insightful comment. So when I was talking about stereotype patterns of branching morphogenesis in the lung, and I mentioned that there is some stochastic loss of branching from individual to individual, and there are some stereotypic losses of branching from strain to strain. Somebody asked me whether this is at all clinically relevant. And the answer to that question is we don't know. But this is an extremely important question in the context of the kidney. And in fact, the kidney is arguably one of the best systems in which it has been observed that differences in the branching morphogenesis program during development can have extremely important implications for adult health. So it turns out that the number of nephrons that you're born with is effectively a term, the term that is used to describe this is called the nephron endowment, is fixed and can be highly varying in an individual, meaning once the animal is born or once the individual is born, there is no more genesis of nephrons that occur. So you're stuck with whatever the number of nephrons that you're born with is. But what is interesting is that if you look across human populations, you will find that the number of nephrons that you're born with can vary tremendously from individuals to individuals. So for example, it can in fact vary over an order of magnitude. So you can have some humans who are born with 200,000 nephrons. And remember, this number is not going to change once you're born. And you can have individuals who have as many as 3 million nephrons. Why is this important? This is important because a long-standing hypothesis is that ultimately the loss of nephrons is what predisposes human beings to hypertension. And there are certain populations on the planet, Australian Aborigines, for example, who are congenitally predisposed to being born with lower numbers of nephrons. And individuals who fall into these populations are predisposed to early onset hypertension as adults. So one of the reasons why a lot of work has gone into trying to understand, for example, the developmental programs of in the kidney is because of this reason. The fact that you're born with a fixed number of nephrons and the number of nephrons that you're born with almost directly correlates with your predisposition to developing hypertension as an adult. And so this is an important point of contrast between our understanding of branching morphogenesis in the lung and branching morphogenesis in the kidney and its relative implications for health. Our understanding of, for example, the extent of branching that occurs in the alveolus is still incomplete in the lung. Is the extent of branching of the alveolus, of the alveolar tree, for example, like the airways? Is it stereotype, or is it more like the kidney where it has some stereotypic elements? And do the number of such alveolar elements correlate with more or less predisposition to disease? I mean, these are the kinds of questions that now arise as a function of sort of making this comparison and thinking more deeply about. So there are two other aspects that I want to sort of go into briefly before I stop. And this speaks to some of the questions that the class more generally posed. So many of you, for example, so the first is an interest in the liver. Why did I not choose the liver, and why did I choose the kidney to talk about rather than the liver? This is one question that I wanted to sort of speak to you about, because I think it'll give you a slightly different idea about organogenesis than what you learned from just comparing the lung and the kidney. And the second question that I want to go to is a question that Utkarsh asked, which is a more detailed mechanistic question concerning the role of the extracellular matrix during organogenesis. So let me address the first question first. Why did I talk about the kidney and not the liver? So it turns out that the liver is again very different from the lung, and it's more similar to the kidney in terms of its mechanism of branching morphogenesis than it is to the lung. So what happens in the liver is very simple. What happens in the liver is that there is a collection of cells which are epithelial in nature that undergo an epithelial to mesenchymal transition. There is a layer of extracellular matrix, and this begins to speak to the question that Utkarsh asked also, that surrounds the distal tip of this hepatic bud, if you will. And so at some point in time, there is an inductive interaction between the subepithelial mesenchyme and the hepatic bud. And these cells that comprise the hepatic bud undergo an epithelial to mesenchymal transition. They invade the basal lamina that surrounds the hepatic bud, and then begin to form what are called cords of epithelial cells, meaning they undergo a mesenchymal to epithelial transition again to create these cords. The reason I didn't talk about the liver and preferred to focus on the kidney is because while the process of hepatic cord formation ultimately leads to a branched organ, which is glandular, meaning it has luminal spaces into which material is secreted and is richly associated with a series of blood vessels, is that in 3 dimensions, our understanding of precisely how this network is formed is very poor. In fact, it's extremely complex, and our understanding of the stepwise progression of the branching morphogenesis of the liver leading, from hepatic cord formation is very poor. And so I figured that if we were to think more in terms of comparing the lung to the kidney, then we might have a more direct correlation between 2 programs of branching morphogenesis that are more or less comparable, and this can speak to the concept of stereotypy and so on. The liver, in contrast, is a much more complicated organ where we don't really understand the pattern of branching morphogenesis simply because its architecture is so complicated. Finally, speaking to the question concerning extracellular matrix, see, this is a bit of a paradox. There is no doubt that if you begin to mutate genes that encode components of the extracellular matrix, that development will stop. In fact, development will stop way before organogenesis begins. The fibronectin knockout, for example, will die well before we begin to talk about organogenesis. But the question that is being asked is, what is the relevance of cell matrix interactions in the context of shaping or developing organ? And our answer to this question is extremely poorly formed. Let's focus on one aspect of the kidney, which I think will be relevant here. There are very few studies that actually speak to an in vivo relevant process of extracellular matrix remodeling in the context of lung development. But there is one aspect, one particular study that speaks to this in the context of the kidney. And let's just look at that and try to see what we can learn from that. So as I mentioned to you, the ureteric bud, the branching of the ureteric bud involves an intimate interaction with the cap mesenchyme, and this interaction involves Wnt11 and GDNF. It turns out that there is a novel component of the extracellular matrix that is produced by the ureteric bud. I believe it's called nephronectin. And there is an integrin that specifically interacts with nephronectin. I believe it's an alpha 8 beta 1 integrin that is expressed in the cells of the mesenchyme. Now, what is interesting is that if you look at both the nephronectin knockout and you look at the alpha 8 beta 1 knockout, you see that in some cases there is kidney agenesis, meaning there is a defect in the formation of the collecting buds, and of course, subsequently, the formation of the adult kidney. I mean, this should now be logically obvious to you because the ureteric bud and its growth is intimately linked to the specification of renal vesicles and the growth of nephrons and so on. So when people looked a little bit more carefully, what they noticed is that in these mutants in which nephronectin or alpha 8 beta 1 integrin were deficient, you found that there was a loss of GDNF at a very early stage during branching morphogenesis in a precise window of time. In other words, if you looked at a slightly later stage, so if you look between, say, 11.5 and 13.5, you found that there was a reduction in the levels of GDNF. But if you looked at later stages, you found that the levels of GDNF came back to normal and that there was some branching that occurred, even though it didn't completely lead to the formation of a normal kidney. So what this suggests is that this ECM interaction, which involves the engagement of an integrin by a specific component of the extracellular matrix, seems to be regulating signaling that is relevant to the growth of the structure. The other, I mean, this is the conservative interpretation of for example, the data that has come from the kidney. If you go to the liver, for example, there the role of the extracellular matrix, it seems, is more structural. In other words, the basal lamina that surrounds the hepatic bud or the organized extracellular matrix that surrounds the hepatic bud, from which cells will undergo an EMT, invade the bud to then lay the foundation for the hepatic cords and branching morphogenesis in the liver. Here, the ECM seems to be forming or seems to be playing more of a structural role in preventing a certain morphogenetic event from happening. So I think if you take a step away from our general conceptualization of the role of the extracellular matrix during organogenesis, what you would find is that all of the examples that exist fall into one of these two classes, meaning either the perturbations of cell matrix interactions are going to perturb signaling that is intimately linked to the morphogenetic process, or you will find that the loss of extracellular matrix components will alter structural aspects of the morphogenetic process, meaning physically prevent cells from either undergoing an EMT or from migrating from an epithelium into the mesenchyme and vice versa. So with that, I'll stop and I'll take any questions.
Hello.
Yeah.
So this mesenchymal to epithelial transition, is this transition very common during development, or
Very,
is majorly seen? Okay.
very. In fact, if you just go back to that initial slide, and you think about how cells are. So you start from the very beginning, inner cell mass to epiblast, MET. Gastrulation, endoderm leaving primitive streak to forming an endodermal epithelium, MET. So if you just try to visualize how these processes are occurring, and then of course, okay, now you formed epithelial ectodermal sheath, epithelial endodermal sheath, mesenchyme. Organization of the mesenchyme into cords leading to, for example, the establishment of the nephric duct, mesenchyme to epithelial transition. I mean, it is everywhere.
Okay.
So, I mean, one of the reasons why I don't like to go into too much detail when I discuss development is because I think it's important at some level for you guys to be able to visualize this process, rather than caught up in all of that nomenclature and details about signaling. Just try to visualize this process. And if you just sit and think about it, you'll find that this is integral to almost every aspect of development at every stage.
Yeah.
Hello. Yeah, I was wondering, we learned now that the ureteric bud sort of induces the mesenchymal cap to start producing these pretubular aggregates. But I was wondering, so is there any sort of special or specific interaction between that ensures that each collecting duct then binds to or merges with the nephron, or how does that sort of ratio maintain?
That's a fantastic question, and there is no clear answer to that question. I think people have identified gene regulatory networks that are critical. I think one thing that I think is important for you to set in mind is that, see, most of the lecture has been cartoons. And if you take a step back and ask the question, what are these cartoons based on? You'll find that they are largely based on genetic evidence, right? Where genes have been deleted, and what you've essentially looked at is a kidney agenesis phenotype, which you've then tried to deconstruct. The kind of question that you're asking requires a far more sophisticated type of analysis, where you can remove gene function in real time and observe the behavior of cells. But I think the bigger question that you're speaking to, which is what is causing these cells to aggregate with one another, show a differential adherence, for example. How does this population of cells transmit from one bud to the next bud upon branching? I think these are deep questions. Even, for example, the question of how the intermediate mesoderm forms in the first place. What causes these mesoderm? So one idea is that, the identity of the intermediate mesoderm, which is the structure that gives rise to the nephric duct, as different from the axial mesoderm or the paraxial mesoderm, whatever that is. This is based on, for example, the timing of exit during gastrulation. But again, how does the transcription factors that are regulating the specification of these cells, leading to their segregation to forming these specific cords, which then undergo reorganization to form epithelia? Open question. Remember, ultimately what you're working with is genetic data where in the absence of a particular gene product, the process fails. So going from that to answering the kind of question that you're asking it requires a lot more work. And it requires some capability to look at this process in real time.
Yeah, I was just wondering if at all, if we had to take a guess, and if we imagine that it is some kind of stochastic process, then is it possible that the ones that don't merge would sort of just degrade, and the remaining ones that did bind are the ones that we get finally?
Yes. It is possible it's stochastic. It's also possible that it's actively driven. So for example, this does not speak to the mesenchyme, but this speaks to a phenomenon that has been observed in the ureteric bud and in many other branch networks. So for example, it's also been observed in the Drosophila tracheal system, which is the system I work with. So when you see so here is what is happening. Nobody understands how this happens, by the way, but it speaks to how segregation is happening. So when a bud is growing, it's very clear that the cells at the tip are slightly different from the cells at the base. What is interesting is that if you now make a genetic perturbation, so that let's say that this bud is growing in response to FGF. If I now make a genetic perturbation such that I remove, say, the FGF receptor in this growing field, what will happen is that the cells that have FGF receptor will converge at the tip. The cells that lack the FGF receptor will naturally fall back. If you start removing components that are relevant to FGF signaling, for example, not the receptor, same phenomenon. Cells that are competent to respond to FGF will congregate at the tip. Cells that fail to respond to FGF will fall into the stalk. So this is not a stochastic process. This is a process that is actively being driven by differences in levels of FGF signaling. So you might argue that these differences could be dependent on stochastic processes, which is a different matter. But it's entirely possible that you're segregating populations of cells depending on, say, differences that may exist in these cell populations in their responses to a specific signal, rather than by a purely stochastic process, if you know what I mean.
Yeah, thank you. Yeah.
Well, if there are no further questions, we can bring the class to an end. I hope this was helpful. If you have any further questions, you can always write and ask me, and I'll be happy to respond. Bye for now.
Thanks, Arjun. Bye.
Thank you.
Bye.
Bye.
Thank you, Arjun.
Thank you. Bye.
Thank you.